Hello everyone! In this video we're going to study elimination reactions. Let me briefly introduce to you the plan of the current lecture. We will begin with the classification of elimination reactions and continue with three main mechanisms of β-eliminations: E1, E1Cb, and E2. We are interested in the rate laws of these reactions and their general mechanisms. Afterwards, we are going to discuss competitions between different elimination mechanisms and between eliminations and substitution reactions. To be more specific, we will study the influence of solvents, leaving groups, substituents, and bases on these competitions. Then, with this knowledge in hand, we will describe the regio and stereoselectivities of E1 and E2 reactions. In the end, we will discuss α-eliminations and some specific examples of syn eliminations in combination with the corresponding mechanism. Let us begin with the definition of elimination reaction. Reactions in which two atoms or atom groups X and Y are removed from a compound are named eliminations. It is important to note that X and Y can belong to the same molecule, or they can be parts of different molecules. On the slide, the first possibility is demonstrated on the top and numbered 1, whereas the second general mechanism is depicted below and is denoted with number 2. You can also clearly see how leaving groups are generalized with capital letters X and Y, which are colored with blue and green respectively. I would like to stress out that both colorings are going to be used during the whole lecture consistently, so you will be able to easily determine which atoms and atom groups are eliminated. Elimination of two bromine atoms from 1,3-dibromopropane mediated by magnesium illustrates the first general mechanism, whereas the hydrohalogenation of iodoethane represents the second one. Furthermore, X and Y can be attached not only to carbon atoms, but also to heteroatoms. Carbon atoms, in turn, can be sp3 or sp2 hybridized. However, in this course, we will consider only sp3 hybridized carbon, as such eliminations are way more popular in organic chemistry. And here, we come to something so special about elimination reactions. Depending on the distance between the atoms or atom groups X and Y, their elimination has a distinct designation. So, if X and Y are attached to the same carbon atom, the elimination is then classified as α-elimination. Parenthetically, there is a useful designation for two substituents that are attached to the same atom. They are named geminal. For instance, let us take a look at the dehydrohalogenation of trichloromethane mediated by sodium hydroxide. The typical product of such a reaction is carbene, neutral electron deficient molecule containing a carbon atom with a valence of 2 and 2 unshared valence electrons. Now, let us consider another case, namely when X and Y are bonded to neighbored carbons. The elimination is designated then as β-elimination. By the way, there is a special name for two substituents which are bonded to neighbored atoms. Chemists call them vicinal. The β-eliminations lead to the formation of pi bond. In order to better understand this type of elimination, take a look at the reaction presented on the slide. Here, you can see the dehydrobromination of 2-bromo-2-methylbutane under basic conditions, which yields as main product isobutene. If X and Y are attached to carbon atoms, which are separated with N carbons, then their removal is named 1N elimination. So, this elimination leads either to the formation of pi bond or to the cyclic product. On the slide, you can see two examples of 1N eliminations. The first reaction above represents the dehalogenation of 1,4-dibromobutene in the presence of metallic zinc. Let us number the carbon atoms: 1, 2, 3, and 4. You can clearly see that X and Y are attached to the first and the fourth carbons. The major product of this reaction is buta-1,3-diene. You're already familiar with the second example of 1N eliminations, which leads to the formation of a cyclic product, namely cyclopropane. I hope the numbering in this case is obvious. As alpha and beta eliminations are much more popular than other types of eliminations, this video will be dedicated to the description and thorough discussion. Now, let us focus on beta eliminations as they represent the nucleus of our today's lecture. What makes up the difference between the beta elimination mechanisms? We can differentiate between three mechanisms of beta eliminations. They differ from each other in the timing of CX and CY bonds breaking. Y is usually a hydrogen atom, and X represents the leaving group. We depicted exactly this case on the slide here. The first possibility which one can think about is to cleave both bonds simultaneously. It means the process is concerted and passes through the single transition state and forms no intermediates. I would like to turn your attention to the fact that concerted means only through one transition state and is not equivalent to simultaneous. There is another term that is responsible for simultaneous, namely synchronous. The difference between concerted and synchronous is not important for us now, and you can read about it in future in any textbook dedicated to physical organic chemistry. As you probably remember, the term concerted we have already discussed in our previous lecture about nucleophilic substitutions. Exactly this general mechanism in curly arrows formalism is depicted on the left part of the screen. In order to better understand the first possibility of elimination, let us take a look at the energy profile of the reaction on the slide. It is obvious that it is a graph, where the reaction coordinate is an independent variable and energy is the dependent one. To be honest, the energy profile of the reaction has a clear mathematical definition. However, we will not discuss it thoroughly in this lecture, as it is the subject of theoretical chemistry. Nevertheless, it is worth noting that all points on the graph, in other words, all chemical structures such as products, reactants, transition states, and intermediates, are uniquely defined in mathematical terms. So, we will use them now for the graph description. For example, there is only one saddle point on the graph, which represents the transition state. There are no local minima, meaning that no intermediates form as the reaction proceeds. Lastly, there are two global minima. The first one represents reactants, substrate and base, denoted with capital B on the slide. The second minimum is products, namely leaving group, olefin, and protonated base BH+. Also, you can see the activation energy of the reaction colored in red and reaction energy in yellow. We are not going to specify whether these energies are Gibbs free energies or enthalpies, because this difference does not play an important role now. We can see that both bonds CY and CX are broken simultaneously. Such a reaction is called E2 bimolecular elimination. I would like to turn your attention to the fact that the energy profile of E2 mechanism is very similar to that of SN2, which we have discussed in the previous lecture. The good example of bimolecular elimination is the dehydrohalogenation of 2-bromobutane mediated by hydroxide anion. There are usually two reactants in eliminations. The first one is base, represented by hydroxide anion in this special case, and the substrate, containing leaving group represented by bromide in case of 2-bromobutane. Hydroxide anion attacks beta hydrogen atom, and as you can see, simultaneously carbon-bromide bond begins to break. The reaction passes through only one transition state, in which both CH and CBr bonds, highlighted in red and blue, are ready to cleave. The reaction yields 2-butene, bromide anion, and a water molecule. 2D representations are sometimes not so easily understandable, but are by far the most popular in organic chemistry, so let us look at them. Now, you can see the two-dimensional representation of the reaction mechanism on the slide. I would like to stress the fact that curly arrows, lone pairs, and bonds in two-dimensional representation are colored consistently with bonds forming and cleaving in 3D. Here, you can also see reactants, transition state, and products of the same reaction. Now you are familiar with the general idea of how the E2 eliminations proceed. I hope you remember that there are two more mechanisms to clarify. Let us begin to consider the second mechanism. Another possibility is to break the CX bond before CH. This process leads to the formation of carbocation intermediate, which reacts further with base, yielding the corresponding compound with pi bond. Such a reaction is called E1 monomolecular elimination, and this general mechanism can be seen on the left. Look at the energy profile of the E1 mechanism on the right part of your slide. You can clearly see the local minimum represented by carbocation. This intermediate is separated from reactants and products by two local maxima, which are transition states. You would ask, why is it important to know? And the answer is clear. The number of transition states defines the number of different activation energies. Obviously, there are two of them differing from each other in magnitude. This difference will be very important a bit later when we will discuss rate laws. Lastly, there are two global minima. The first one represents reactants, substrate and base, denoted with capital B on the slide. The second minimum is products, namely leaving group, olefin, and protonated base BH+. If you compare the energy profile of this mechanism with that of SN1 reaction, you will find out a lot of similarities. As usual, the reaction energy is depicted in yellow and activation energies in red. You can see a typical example of this mechanism, which is the hydrobromination of third butyl bromide mediated by water. The first step is represented by heterolytic cleavage of the CBr bond. The reaction passes through the first transition state and yields bromide anion and tertiary carbocation, quite stable intermediate. The stabilities of different carbocations have been also clarified in the lecture dedicated to nucleophilic substitutions. The second step of E1 mechanism is a proton transfer between base and carbocation. Obviously, water acts as a base in this case. It attacks beta-hydrogen atom and abstracts proton passing through second transition state and yielding isobutene and hydroxonium ion. On the screen, you can see the products, whereas bromide anion is omitted for clarity. Now you can see the two-dimensional representation of the reaction mechanism on the slide. Till this point of time, we have discussed two different timings of CX and CY bonds breaking, so there is only one possibility left. The last option is very similar to the previous mechanism. However, it differs in the sequence of bond breakage. First CH bond is broken, which leads to the formation of a carbanionic intermediate. The second step is represented by the CX bond break, which leads to the formation of pi bond and anion. Such a reaction is called E1CB, elimination from the conjugate base, because X is eliminated from the carbanionic intermediate, which is the conjugate base of starting material. As usually, the general mechanism of this reaction is depicted on the left. The abbreviation EWG means electron withdrawing group. We will discuss it in detail a bit later in this lecture. Let us briefly examine the energy profile of this mechanism, because it is the only way to understand the kinetics later. It can be found on the right. We can see that it is very similar to that of the E1 mechanism. The main difference is the negatively charged intermediate and the opposite sequence of CX bond cleavage and abstraction of the proton by the base. Furthermore, the activation energy of the first step is smaller than that of the second one. Several reactions follow this type of elimination mechanism. For instance, the formation of acrylane from beta-hydroxypropanol mediated by hydroxide anion. During the first step, proton is abstracted by hydroxide anion, passes through the first transition state, and yields stable negatively charged intermediate, enolate, and a water molecule. Now you can see better the structure of enolate. The second step of E1CB mechanism is heterolytic cleavage of COH bond, which proceeds via second transition state and leads to the formation of neutral stable acrolein and hydroxide anion. The structures of products can be better seen now, where the water molecule formed in the first step is omitted for clarity. The two-dimensional representation of the reaction mechanism is depicted on the slide. Ultimately, here is a brief summary of all three beta-elimination mechanisms so you can explore the key differences between them. As you can see, the names of the beta-elimination mechanisms also include numbers, the same pattern you have learned while dealing with nucleophilic substitution mechanisms. But what do these numbers tell us? These numbers indicate the molecularity of the rate-determining step. The rate-determining step is the slowest step of a chemical reaction that determines the speed at which the overall reaction proceeds. For reactions that include more than one elementary step, the rate-determining one is that with the highest activation energy. For E1 mechanism, the rate-determining step is represented by dissociation of Cy bond, because the corresponding activation energy is much higher than that of the second step. You have noticed that only one compound is involved in this rate-determining step, meaning that this step is monomolecular. The subsequent abstraction of the proton is much faster and does not affect much the overall reaction rate. So, the reaction rate depends only on the concentration of the initial substrate and does not depend on the concentration of the base. The rate law of E1 mechanism is shown on the slide. As you can see, the rate of the reaction RE1, which proceeds via E1 mechanism, depends on the rate constant denoted as Ke1, and concentration of the substrate R containing leaving group X. The corresponding units are represented below. And what about the E2 mechanism, you would say? For E2 mechanism, the rate-determining step is represented by dissociation of CX bond and simultaneous abstraction of the proton by a base. In other words, two compounds, namely base and substrate, are involved in a single reaction step. It means that the rate-determining step is bimolecular. So, the reaction rate depends in this case on the concentrations of the initial substrate and on the concentration of base. The rate law of E2 mechanism you can see on the slide, of course with corresponding units. Now, let us discuss the kinetics of elimination from the conjugate base. The conjugate base is the carbanionic intermediate formed in the first reaction step. Number 1 indicates the monomolecular character of the rate-determining step, dissociation of CX bond in the previously formed carbanion intermediate. The abstraction of the proton during the first step is much faster than CX bond cleavage. In contrast to the E1 mechanism, it has a direct influence on the overall reaction rate because it defines the concentration of carbanion, which is involved in the rate-determining step. To be more specific, the concentration of base is directly proportional to the concentration of carbanion, which is involved in the rate-determining step. On the slide you can see the derivation of the kinetic equation for the E1 CB mechanism. Let us evaluate it together. First, notice that proton abstraction is reversible, which means we are dealing with equilibrium. It makes it possible to formulate the equilibrium constant, which depends on the concentrations of reactants and products. After that, we can rewrite the equation with respect to carbanion concentration. Lastly, we can substitute carbanion concentration in the rate law with our new expression. It is quite complex compared to the rate laws of E1 and E2 mechanisms. In some cases, we can unite the first two multipliers to one and name it constant. We will not describe the reasons for it because it's not a subject of our today's lecture, but of physical chemistry. That is why actually both concentrations are included in rate law, despite the monomolecular character of the rate-determining step. I hope it is clear for you. If not, you are welcome to ask the questions in the comments. Summing everything up, we have learned the general mechanisms and rate laws of beta-eliminations. But how do we know which of these mechanisms does the reaction follow? Well, which factors do we need to consider to correctly predict the mechanism? In order to elucidate key factors, let us have a look at the general scheme of the reaction. As you can see, there are two major reactants, substrate and base. So the first factor is obvious, base. So let us examine the second factor a bit more precisely, namely substrate. It contains the leaving group, which we have formally denoted as X. Let us add it to the list of key factors. As we have already discussed, during reactions that follow E1 and E1Cb mechanisms, corresponding intermediates are formed, carbocations and carbanions. It is obvious that substituents play a key role in the stability of these intermediates. Let us add them to the list. We have already seen in the previous chapter dedicated to nucleophilic substitution how solvation can stabilize leaving groups and intermediates. It is clear that now you have learned quite a lot of new information, so let us have a short break. During this break, we'll find an answer to the question, why E1 mechanism is so important for all of us and how the nature uses it. Let us have a look at histidine. L-histidine is a proteinogenic amino acid with unique biochemical and physiological properties. Proteinogenic means that it's used in biosynthesis of proteins. The unique chemical properties of histidine, which are mainly attributed to the imidazole ring, include proton buffering, metal ion chelation, and antioxidant activities. Moreover, histidine is nutritionally essential for human beings because it is not synthesized in our bodies. In order to get this amino acid, we need to digest histidine-containing proteins. And now you would probably ask, if we and other animals do not synthesize histidine, then which living organisms can do that? The answer is simple. Prokaryotic organisms such as well-known E. coli and plants can synthesize histidine. We are not going to study in detail the complete biosynthetic pathway as it contains 10 steps and it is quite complicated and time-consuming. On the other hand, we'll focus on the single step, namely 6th one. It is the conversion of d-erythroimidazole glycerol phosphate into imidazole acetal phosphate catalyzed by enzyme imidazole glycerol phosphate dehydratase. Phosphate is denoted as OP. From the name of enzyme, it is obvious that it is catalyzing dehydration reaction. Histidine biosynthesis is a target for herbicide and antibacterial agents, with imidazole glycerol phosphate dehydratase a key enzyme within this pathway. As a result, IGPD was the focus of inhibitor design programs with several potent herbicides in development. Some of the potent inhibitors, such as IRL 1695 and IRL 1803, are now presented on the slide. Before inspecting the mechanism, let us have a look at this beautiful enzyme. It is of octahedral symmetry and contains 24 subunits and more than 33,000 of atoms. You can see the mechanism of dehydration process on the screen. During the first step, the resonance-stabilized carbocation is formed and OH leaves as hydroxide anion. It is quite an unusual situation when OH group acts as leaving group in E1 mechanism. The formerly discussed enzyme traps hydroxide anion with acidic proton colored red, thus leading to formation of water molecule. Then proton in beta position is abstracted and new pi bond is formed. Finally, the product is tautomerized to imidazole acetal phosphate. To my mind, this reaction serves as a perfect example of E1 mechanism in biology, and it could be important for agriculture and pharmaceutical sciences. Now, it is time to finish the break and return to our main topic of the discussion. Now, I would like to go to the next part, where we will be discussing factors that favor the monomolecular substitution mechanism E1. Look at this graph. The reaction here proceeds via an intermediate carbocation, so the more stable carbocation is, the better it is for the E1 mechanism. We have already learned in the previous chapter how the stability of carbocation depends on its structure. The least stable is methyl carbocation followed by primary carbocations. These two groups are so unstable that do not literally form in the solution. According to the scheme presented on the slide, secondary carbocations are more stable than primary, but significantly less stable than tertiary. It means that secondary alkyl halides also literally never follow E1 elimination mechanism. In contrast, tertiary halides are the best substrates for the E1 mechanism. Until this moment, we have discussed only alkyl carbocations. However, there are other types of carbocations. For example, pi conjugation is quite an effective way of stabilizing carbocations. Such conjugation enables to formulate more resonance structures, for example, for allylic and benzylic carbocations. That's why allylic carbocations are less stable than tertiary alkyl carbocations, but more than secondary. And benzylic is the most stable among the discussed carbocations. It means allylic and benzylic derivatives can follow E1 elimination mechanism. In order to form the pi bond, both benzylic and allylic carbocations should be substituted. For example, benzylic alcohols follow E1 elimination mechanism under strong acidic conditions, like in the presence of sulfuric acid, and yield styrene or its derivatives. The dehydrohalogenation of dimethyl substituted allylic bromide and methanol, which results in the formation of diene, represents the E1 mechanism for elimination from allylic derivatives. Another way of carbocation stabilization is heteroatom in alpha position, which is capable of free electron pair donation. It also makes it possible to formulate more than one resonance structure, thus stabilizing the whole system. You can see examples of such substituents on the slide, such as chlorine, bromine, O-methyl, NME2, etc. That is why we can add such carbocation to the list and put a tick nearby. The effect of the leaving group is obvious. The better it is, the faster carbocation is formed, thus favoring E1 mechanism. We have already learned the comparison scheme of various leaving groups in the previous chapter, so we will not put much emphasis on it now. If you want to refresh your knowledge, you can briefly examine the scheme presented on the slide. We have already said that solvent can also have a significant influence on the mechanism according to which the elimination will proceed. And again, as we know, protic polar solvents favor SN1 reaction because they can solvate the leaving anion properly, thus decreasing its energy. The same trend can be observed in the case of E1. Polar protic solvents such as alcohols, carboxylic acids, and water-based mixtures stabilize the leaving group and the forming positively charged intermediate, carbocation, because they represent good ionizing, ion-solvating media. That's why they favor E1 mechanism. There are lots of polar protic solvents, some of them can be seen on the screen. The most acidic protons are colored red. The numbers below the structures are corresponding dielectric constants. The higher the number is, the more polar is the solvent. The last factor to discuss is base. Being more specific, its concentration and pKb value. If one examines the rate law of E1, he will find out that base concentration is not a part of the equation. The concentration of base does not affect the rate of E1 reaction, so it cannot favor or disfavor it from first sight. Further investigation of competitive E2 mechanism, however, will show completely other dependence. The higher is the concentration of base, the higher is E2 reaction rate. That's why high base concentrations favor E2 mechanism and thus indirectly disfavor E1. Let us evaluate the influence of base strength. Look at the reaction presented on the slide. Alkene is protonated with BH+, yielding carbocation and conjugate base B. It is quite complicated to protonate alkene. In order to do it, you need to use some strong acids such as hydrobromic acid. Do you remember Bronsted acid-base theory from your inorganic chemistry course? One of the key postulates of this theory is that strong acid has a weak conjugate base and vice versa. On the slide, the weak base is alkene, so the conjugate acid, carbocation, is a strong acid. Consequently, carbocation formed during E1 mechanism will give its proton to any particle, which can act as a base. In the solution, the probability of collision with solvent is much higher than of collision with any other base, so carbocation in E1 usually reacts with solvent. That is why the strength of the added base is not important and does not favor the E1 mechanism. In the next video, we are going to discuss the competition between SN1 and E1, so you will better understand why this mechanism is not well suited for synthetic applications. And here again is a brief summary of factors favoring E1 mechanism. Stabilized carbocation, good leaving groups, polar protic solvents, and small base concentrations. I hope this part is clear for you. If not, you know where the comments are. Now let us turn to the E2 mechanism and discuss it in more detail, the factors that favor it. The same discussion about E1 mechanism we have started with the analysis of the substrate structure, so we will follow more or less the logic of the subsequent discourse. We concluded that E2 is a concerted mechanism, which means it proceeds via a single transition state and does not lead to the formation of any intermediates. In contrast to E1 mechanism, where the stability of carbocation had a positive influence on the probability that reaction will follow this mechanism, the opposite is true for bimolecular, ChemU elimination. The more unstable is the possible carbocation, the less is the probability of its formation, so the higher is the probability of E2 reaction to occur. For instance, methyl and primary alkyl halides react only in E2 fashion as the corresponding carbocations are too unstable. The secondary alkyl halides can follow both mechanisms. In order to correctly predict mechanism for them, other factors should be taken into consideration, namely base strength. The same is true for tertiary alkyl halides. If we compare the rates of E2 elimination for tertiary, secondary, and primary alkyl halides, we will notice that reaction rates decrease in this row. It might be confusing since we are used to steric hindrance having a negative influence on kinetics. The bulky substituents usually decrease the rate of the reaction, as they significantly decrease the accessibility of the reaction center. In other words, they are making the interaction between reactants less probable. However, tertiary halides react faster than secondary, and secondary faster compared to primary. There are two main reasons for that. First, the bases are not as sensitive to steric hindrance as nucleophiles because protons that should be abstracted are small and usually on the outsides of the molecules. Second, the more highly substituted alkene is, the more stable it is. It is supported by thermodynamics of hydrogenation reactions of different alkenes presented on the slide. So the higher is substitution grade, the less exothermic hydrogenation reaction is. The same trend is also valid for corresponding transition states. The higher is substitution, the more stable is the transition state. According to the well-known Arrhenius equation, the more stable the transition state is, the bigger is the rate constant, so the reaction proceeds faster. The effect of the leaving group is the same as for E1 mechanism. The better it is, the faster CX bond is cleaved, thus favoring E2 mechanism. It is important to note that good leaving groups often represent weak bases, and they should be weaker than bases initially used for elimination reaction. The influence of solvent is directly linked to the base strength and its effect, so let us study the effect of the base prior to that of solvent. We have already mentioned that an increase in base concentration favors the E2 mechanism as it is present in the rate law. The pKb value of the used base in contrast to the E1 mechanism is also important for bimolecular elimination. To be more specific, bases with low pKb favor the E2 mechanism. In other words, if we use a strong base for elimination reaction, such as presented on the slide, we definitely deal with the E2 mechanism. The last factor to evaluate is solvent. chemists have noticed that change from polar protic to polar aprotic solvents significantly increases the base strength. This happens since the base is surrounded by a huge solvation sphere in protic solvents. The solvent molecules are capable of building strong hydrogen bonds with the base. The hydrogen bonds are depicted with dashed lines and the bases usually with capital B. For instance, let us take a look at the solvation of ethoxide with the water molecules on the slide. It makes the base more stable and thus less basic, which has a negative influence on the probability of the E2 mechanism. In contrast, polar aprotic solvents such as acetone or dimethyl sulfoxide do not form hydrogen bonds with solute, in this case base. Solvation spheres held only with dipole-dipole interactions and van der Waals interactions, which are much weaker and thus easier to break. It leads to the increase of the basicity. Simply speaking, polar aprotic solvents enhance E2 mechanism and sometimes can lead to the change from E1 to E2 elimination by a significant increase of basicity. But what does it mean for us? It means that the correct prediction of elimination mechanism for secondary and tertiary halides implies first analysis of base strength and secondly the influence of solvents on the base strength. And here again is a brief summary of factors favoring E1 mechanism. All in all, it is the unstable carbocations, good leaving groups, high base concentrations, strong bases, and polar aprotic solvents which favor E2 mechanism. Now it's time for the second little pause. During the last few minutes we have discussed bimolecular eliminations. I would say that this mechanism is the most popular in organic chemistry among all elimination mechanisms. However, it is quite complicated to find any biochemical reactions which proceed according to the E2 mechanism. It is rare, but still possible to find an example of how nature uses it, for instance, in our bodies. Before discussing the specific reaction, ask yourself, have you ever heard about the citric acid cycle? To my mind, the majority of people who are watching this video have heard these words at least once. Nevertheless, I would like to remind you, the citric acid cycle is the central metabolic hub of the cell. It is the gateway to the aerobic metabolism of any molecule that can be transformed into an acetyl group or dicarboxylic acid. You can see the complete cycle on the screen now. The cycle is also an important source of precursors, not only for the storage forms of fuels, but also for the building blocks of many other molecules, such as amino acids, nucleotide bases, cholesterol, and porphyrin, the organic component of heme. The citric acid cycle, in conjunction with oxidative phosphorylation, provides the vast majority of energy used by aerobic cells, in human beings greater than 95%. It is highly efficient because a limited number of molecules can generate large amounts of NADH and FADH2. As usually, we are interested not in the whole cycle, but in one specific reaction, namely the conversion of citrate into isocitrate, mediated by enzyme aconitase. The reaction proceeds by an intermediate named cis-aconitate. The reaction scheme is now presented on the slide. You can clearly see that both reaction steps are reversible. The one is dehydration and the second is the addition of water. I would like to stress the fact that both eliminations of water from citrate to cis-aconitate and from isocitrate to cis-aconitate are proceeding via E2 mechanism, what is proved by stereochemical outcome. Look at this beautiful enzyme in general, and the reaction center especially, where the isocitrate is bound. The strange orange-yellow cube is the iron-sulfur cluster Fe4S4, which is essential for dehydration because it binds the leaving hydroxide anion. Let us clarify what is happening here. The white dashed line represents the forming iron-oxygen bond. The blue dashed line is the cleaving carbon-oxygen bond, and the yellow one is cleaving hydrogen-carbon bond, and red one is forming oxygen-hydrogen bond. The reaction yields cis-aconitate, and we have already discussed. Now you can inspect the two-dimensional representation of the mechanism. The citric acid cycle is one of the key metabolic pathways in aerobic organisms. That is why inhibition of its enzymes leads to fatal consequences. The well-known inhibitor is sodium fluoroacetate. It is used as a rodenticide in many countries, for example in Australia, New Zealand, Mexico, Japan, etc., to protect plants from rats. It also helps to control the population of possums, deer, and rabbits. Sodium fluoroacetate binds tightly aconitates, thus leading to the increase of citrate concentration in blood. Furthermore, it allosterically inhibits PFK1, the key enzyme of glycolysis. As a result, cells cannot metabolize carbohydrates and produce energy. Lastly, I would like to mention that it is also poisonous for humans. Now, it is time to finish the break and return to the factors that favor the E1CB elimination mechanism. I hope that you are alright so far and ready for the last part for today. We're almost done. The last mechanism that we should discuss is obviously E1CB, monomolecular elimination from conjugate base. I hope you are already used to the logic and sequence of factors that we are going to evaluate. The first step of this mechanism is represented by the formation of intermediate carbanion. It is formed due to heterolytic dissociation of CH bond. The last step is just the abstraction of a proton which yields pi bond. Do you remember? We have discussed previously that the rate law of E1CB mechanism proposes the dependence of rate from concentrations of base and substrate because the base is responsible for the formation of intermediate carbanion. The higher its concentration is, the higher is the rate. It means that carbanion stability plays a crucial role in E1CB mechanism. You have definitely noticed the similarity to E1 mechanism, where we have discussed the stability of carbocation. Let us take a look at factors that influence the stability of carbanions. The first factor is obvious, the number of adjacent carbon atoms. In this case, the more highly substituted carbanions are less stable compared to those less substituted. In other words, methyl carbanion is more stable than primary, the primary is more stable than secondary, and tertiary are least stable. It is clear that the order presented on the slide is opposite compared to that of carbocations, and it is simply because of the positive inductive effect plus I of alkyl groups, which causes the increase of electron density on central carbon, thus decreasing its stability. The positive inductive effect is depicted with dashed blue arrows. Now we have understood the carbanion stability is vital for E1CB mechanism. But how can we then stabilize carbanion? Substituents with -I effect, and of course substituents with negative mesomeric effect -M, have a positive influence on carbanion stability. The first group can withdraw electron density from central carbon, thus increasing its stability. The negative inductive effect is depicted with dashed red arrows. Some examples of substituents that possess -I effect are represented on the slide, for instance, halogens. The second group can efficiently delocalize the charge, thus stabilizing the whole system. In addition, -M substituents allow the formulation of more resonance structures, what also enhances the stability of the corresponding carbanion. On the slide, you can see the typical example how NO2 possesses -M effect and the resulting resonance structures. In addition, some typical -M substituents are also given. Summing it up, electron withdrawing groups stabilize carbanion and thus favor the E1CB mechanism. If they are not present in substrate structure, then elimination from conjugate base does not happen. The last factor to discuss is leaving group ability. Previously, we have seen that better leaving groups favored both E1 and E2 mechanisms. It was so due to the fact that CX bond cleavage was included in the rate determining step and thus higher leaving group ability favored both mechanisms. In the case of the E1CB mechanism, this statement is only partially true. Even though CX bond cleavage represents rate limiting step, the good leaving groups disfavor E1CB mechanism. This, from the first sight counterintuitive trend, agrees with our previous knowledge. E2 mechanism competes with E1CB mechanism, so good leaving groups favor simultaneous cleavage of CH and CX bonds, thus enhancing E2 mechanism. In the case of poor leaving groups, CX bond is cleaved slower than CH bond, so making the formation of the carbanion intermediate possible. Lastly, electron-withdrawing leaving groups, such as fluorine, can stabilize carbanion and thus enhance E1CB mechanism. We have already discussed before that E2 and E1CB mechanisms compete. Also, calculations suggest that the activation energy of E2 mechanism is generally lower than that of E1CB by 30 to 60 kilojoules per mole, which is quite a substantial amount. That is why E1CB is quite rare. Now, let us examine once again an example of E1CB mechanism, acrylene formation. It is important that in this case OH left without prior special activation, for example, tosylation or protonation. It is quite an unusual case when OH plays the role of leaving group without any activation. For instance, it does not happen in nucleophilic substitution mechanisms or in E1, E2, but in E1CB it is possible. Ultimately, E1CB is quite a rare mechanism that competes with E2. Carbanion stabilizing substituents and electron withdrawing poor leaving groups enhance elimination from conjugate base. As usual, here is a brief summary of factors favoring E1CB mechanism. All in all, it is the stabilized carbanions, poor electron withdrawing leaving groups, and high base concentrations that favor E1CB mechanism. This was the last topic for today. Let us make a small recap of what we have learned. We have begun with the classification of eliminations and supported them with examples. Then we have defined the elimination reaction and proceeded further with a thorough discussion of the most important elimination mechanisms, which differed in the timing of the CX and CH bond cleavages. We have evaluated the general mechanisms of E2, E1 and E1CB, and afterward inspecting corresponding energy profiles. This gave us enough information in order to clarify their rate laws. In the last part of today's lecture, we have discussed in detail which factors and why favor each of these mechanisms. In the second part dedicated to eliminations, we will elucidate the competition between elimination and substitution reaction, followed by a detailed discussion about regio and stereoselectivities of E1 and E2 reactions. In the end, we will learn about alpha-eliminations and some specific examples of syn eliminations in combination with corresponding mechanism. On the last slide, you can see the summary of today's lecture. We have discussed everything here, so you can read and learn it by yourself, so you will be able to understand your own weak and strong parts regarding today's lecture.
That's it for now. I would like to remind you that if you have any questions related to this video or any topic in chemistry, biochemistry or pharmaceutical sciences, you are more than welcome to ask them on our website or in the comments below. Don't forget to give us a like and subscribe to our channel. Thank you very much for your attention, and we'll meet once again in the next video.